Transcription factors are proteins that bind to DNA during transcription. The factors have many functions such as controlling the rate of transcription and regulating the amount of protein and RNA available to the cell. Transcription factors have a DNA binding domain, a signal sensing domain, and a transactivating domain. The DNA binding domain is where the transcription factor attaches to certain points on the DNA helix. The signal sensing domain receives signals from the body and broadcasts signals to other transcription factors to regulate how many factors are needed. The transactivating domain contains binding sites for transcription co-regulators that control whether the genes are on or off. Transcription factors control a big part of gene expression. Genes in humans usually begin in the off state. Transcription factors turn them on. When transcription factors bind to DNA, they promote or block the transcribing enzyme. This means that some genes are expressed while others are not. Insulators, made from the CTCF protein, prevent enhancers from binding to DNA. This doesn't allow for the gene to be expressed. Methyl groups prevent insulator proteins from binding to the DNA and allows RNA polymerase to bind to the DNA. This allows the genes to be expressed.
Don't believe me, just watch.
MicroRNA is a small piece of RNA that is usually made in the nucleus and exported to other parts of the cell. At first, they are double-stranded, but are later cut into small parts by dicer proteins and split into one strand by an argonaute protein. These proteins are part of the RNA-induced silencing complex. Because microRNA might need to be used on multiple strands of mRNA, it usually has complementary base pairs on some, but not all target mRNA.
Don't believe me, just watch. Hey, oh!
Epigenetics are the study of the molecules that manipulate the activation of DNA. This study explains why identical twins look the same right after birth and can grow up looking completely different. We will be focusing on two chemical processes that affect the activation of genes. Methylation occurs when methyl molecules bind to DNA and inhibit or activate certain gene functions. They can cause the histones, which contain genetic information, to coil up and make activation and replication of a specific trait more difficult. Acetylation is the addition of an acetyl group that activates a certain gene and makes replication easier. It unwinds the histones, helping RNA polymerase and transcription factors access the strand of DNA and make the replication process easier. The main reason there are differences between identical twins is the environmental differences that affect the activation of certain genes. For example, a rat pup who is raised by a low-nurturing mother does not activate a stress-relieving gene, while another pup who is raised by a nurturing mother is easily calmed after stress. Overall, epigenetics play an important role in explaining why different genetic traits are expressed throughout our lives.
Uptown, funk you up. Uptown, funk you up.